We have seen many interesting clinical trials, but basically clinical trial is a human type of experiment. So therefore, there's a many limitation, restriction, some hope, but patient potentially suffer. Also, investigator could also suffer because we only give medicine to everybody, for example. So why we have to randomize? Patient come here for survival. But we can't do that because this is a very important step to have the medicine available to everybody. Same thing, pharmaceutical company, you might think, oh, they're waiting for the $1 million coming into the company to pay off the investor, but they do also have suffer. They keep negotiating with the FDA and other regulatory authority to protect the patient, to shorten the time of suffer, and to get the medicine available to everybody. So therefore, this is like a more joint venture. Both sides is going to suffer, and both sides have to work together. So that's a clinical trial. So that I want you to understand that before going into the explain everything. And also we are kind of assuming that everybody understands phase 1, phase 2, phase 3. So, but I'm going to spend some time to explain that. Before I go into the human trial, investigator, basic researcher find some signal by using a cell line, this medicine should work. I believe my life is on this chemical, so this should work, and I have to move in to the next stage. They have spent a lot of time, effort, and then find out how to prove that one medicine is important. Then they have to prove that the medicine is work for animal, usually mouse, sometimes rat, sometimes chimpanzee, sometimes dog. So before actually giving this to the patient, they have to make sure there is no toxicity and there is potential efficacy. So there are very long time to develop one medicine. And then once medicine is confirmed to be safe and potentially efficacious, then we are going to start so-called phase 1. Nowadays, phase 1 is called phase 1A, phase 1B. This is like from mouse to human, but still very experimental. So most of medicine that work in mouse doesn't work in human. So therefore, we have to make sure also some unexpected side effect, like a thalidomide. It was not expected in the mouse study. So therefore, we have to start from like a scratch. Then they choose a dose from the mouse experiment, and so-called the phase 1A, so-called dose escalation. So they start from the lower dose and step by step increase. Then they will find out so-called the maximum tolerated dose. So that is a phase 1. So therefore, each patient has a different dosing. Small cohort, 3 patients to 6 patients is getting a medicine to make sure that is safe to be given. And then at the same time, they are looking for some signal that this medicine is going to work. Then they have to move on to either 1B or 2A, which means increase the number of the specific cohort, for example, eye melanoma, skin melanoma, colon cancer, or lung cancer, which has some signal from the animal study, also phase 1A. They are going to increase the number to see whether that is real or not. So that increased the number from like 10 to 20 patients. Then sometimes that 1B is going into the 100-patient clinical trial. Nowadays, they try to save the time, to money, and to make the medicine available to the patient as soon as possible. So then once they finish the phase 1B or 2A, they are going to extend the trial to check some efficacy. Safety is confirmed. This medicine is probably tolerated in the majority of patients, so therefore we're going to try that medicine for the big population. Not biggest, but try to see any efficacy. So then once confirm it, now agony come. So FDA never allow medicine to be approved as a single arm. We have to compare the new medicine with standard care or previously approved medicine. So phase 1 is okay. Everybody nobody, not whether it's work or not, but everybody can get the medicine, no sugar pill. However, if you go to the phase 3, you have to show the superiority, efficacy compared to the standard care. And then problem for the eye melanoma is there's no standard care. So therefore, they have to choose one of the so-called standard care, such as chemotherapy, which is terrible choice in general, and or, for example, immunotherapy medicine, which is almost 0% response rate. So then they have to compare that, or they have to use a placebo arm. Patient doesn't know, doctor doesn't know, but they just keep giving it, and then open up the box. Oh, this patient is getting it. So patient is going to suffer, and investigator is going to feel very bad because they are our patients. We don't give a best treatment, but this is a process to go through. Otherwise, this medicine is not going to be available to anybody. That's a very important process, but it's going to be suffered. So the company tried to choose the best alternative, and then company tried to minimize or shorten the time for the so-called control arm. So that is a crossover model. Sometimes set up the very short interval like a goal. So I want you to understand, if you drop off, okay, I'm going to going to A and B, and then I'm in the placebo arm. I don't want to do it. This is my life. Then a trial is going to be ruined. That's the issue. And then if I don't want to stop medicine, I will just continue forever because that is my health, my life. Now we don't have any conclusion. So the study design is carefully discussed and approved by the IRB, FDA. So if you go into the different route, or if you withdraw, all of a sudden that trial has a serious issue. So we don't need this kind of concern for the last 10 years because there's nothing available. So now there is 7 medicine for skin melanoma. All skin melanoma patients went through the same suffer. They have to compare with GB100 peptide vaccine. We know that it's not going to work. To compare to ipilimumab, we have to compare DTIC with something new medicine because DTIC is approved by the FDA. So now, for the first time in the history, UVA melanoma patients start getting into that kind of dilemma. So, but I want you to understand that is a process to get the medicine available. And the FDA is very sensitive to that. So therefore, they are changing the rule. They are conditionally approved. Start giving a medicine to everybody, but you have to do the final study, kind of stuff, like a more accelerated approval. And then eye melanoma is considered to be orphan disease. So therefore, approval is so quick. However, still we have to go through the traditional rule to confirm that this new medicine is better than traditionally available standard care. So that's a kind of a, suffer. However, I hope that one of these medicines goes through that, and also with our collaboration with the patient, the family, we just make this medicine available by insurance companies quickly. So that's a kind of my message. Also, debate is kind of started, and then you say, this is my life. I don't want to do that. That's true, but that is a reality. And that organ investigator try not to touch that issue, but I need to touch it because this is coming. There are maybe a couple of phase 3s coming. So most of them are placebo control or standard care control. You have to understand that you are the one of the patient also, but you're not patient to help the future patient, So that I wanted to understand that. Anyway, since Dr. Nitsky and mentioned most of these studies, so I will just quickly go down this list. Jefferson is very active in conducting a new clinical trial. And then we have one adjuvant trial, which is Sutent compared to valproic acid. We're going to explain that a little bit more. We have 50% accrual achieved, so therefore we have like a 40 more patients. And then there's a couple of phase 1 trial. One is, as Dr. Latsky mentioned, IMC GP100 study. This is very interesting. I'm going to explain it a little bit more And then we are finishing up the microsphere, radiosphere treatment. I'm going to talk about that later a little bit. And then another one is the Sudelcast study is going to start here also, which has been mentioned, so I'm going to skip it. In coming trial, as Dr. Latsky mentioned, we are going to start DP100 study as a phase 2. That is a kind of phase 3 design. 1/3 of patient is not getting medicine. Okay, and then second one is Dr. Minor on the West Coast. I want to do the clinical study combining a radiosphere plus ipilimumab and nivolumab, standard checkpoint blockade combination. So this is unfunded right now. So, but as soon as we get some funding from somewhere, we're going to start. And then the last one is so-called the PALX51107. This is a BET inhibitor, which somewhat attack on the MIC amplification pathway. So that is very interesting. This is in collaboration with Columbia, Dr. Schwartz and Dr. Kavaho group. Oops. Here you go. Why Sutent? This is like almost 10 years ago history. At that time, Dr. Haber didn't present BAP1. We only know that tumor cells tend to express c-KIT. c-KIT is a growth factor receptor, and then this sunitinib is approved for kidney cancer, which block the c-KIT pathway inhibitor. So therefore, we decided to try that. And then it's not very effective for the stage 4 disease, metastasis patient, but we know that most of the patients have a stable disease. So therefore, we decided to start using it on as needed basis for the very high risk patients. Basically, we cut the dose by 50%, 25 milligram, and for 6 months, and then after that, just follow. So this is kind of design of the current trial. But we only include the patient who has more than 50% recurrence rate, like a monosomy 3 AT amplification, monosomy 3 and a large tumor, as Dr. Haber said, class 2. So then this is a kind of single arm. We only have a red arm. So then a problem is, how can I say this is good? So we have been criticized. You can't use a historical control from paper because different patient population. Therefore, we went into the Wills Eye Hospital to carefully find a patient who is matched to the patient that we have treated. In other words, these are patients from in the same institution and same risk, and blue line is just observed, and red line is patient who receives Sutent. So later on, we found that Sutent suppressed the angiogenesis. So very strong anti-angiogenesis medicine that is required for the patient tumor development. Also, later on, again, Sutent found to suppress the myeloid-derived suppressor cells. Suppression of suppression is positive. So this tumor microenvironment, tumor is attracting many bad cells. So Sutent is kind of blocking it. So we have a couple potential, hypotheses that this medicine may be working. So, and then this is another thing. Okay, Dr. Haber keep talking to me. Okay, why don't you use a HDAC inhibitor? But it's kind of expensive, and then we don't get any approval from insurance company. So therefore, we decided to use a valproic acid. In fact, Dr. Haber showed that this yellow line is the just control, and green line is valproic acid treatment. This is a mouse which received the valproic acid, on a regular basis. So therefore, I decided, and also problem is many primary doctor or oncologists are just giving medicine because this is available from pharmacy. Nobody knows whether it's useful or not. So we decided to include this arm as a second arm. So then the current arm is two randomized two-arm study. If patient randomized to sunitinib, Sutent, 25 milligrams, 6 months. If patient randomized to valproic acid, they are going to take 6 months. Why 6 months? Nobody knows. But usually Sutent tolerate only 6 months, And valproic acid, we don't know how long they can tolerate, but we decided to just stop the medicine 6 months to see whether any signal is coming. If the valproic acid is same as survival, we might use valproic acid because it's cheap. But on the other hand, if sustained is still needed, we might have to talk to the insurance company. So this already mentioned that this is a phase 1. Phase 1 is everybody getting medicine, okay? But nobody knows whether it's work, nobody knows how toxic. So we are escalating a dose. This is a kind of important figure. I forget here. This right side is T cell attaching to the cancer and then shooting the toxic granule. So we try to make a name, and then somebody say immunological magnet. I say gorilla glue. So put them together. They cannot detach because the T cell portion, T cell receptor portion is very highly specific. They don't bind to anything else. In a very small molecule number, like 60 microgram, it is 1,000 times or 100,000 times lower than antibody. So they stick and then don't detach. So what's happened? This is one of the patients who received this medicine. Since skin express GP100 molecule. So therefore, this GP100 medicine go to the skin because they see the GP100 on the cell, normal cell. So as a result, 6 hours, no infusion reaction, 6 hours, clock chinned, patients having fever, skin rash. So that we feel much better because, this medicine is not the water. It's actually working on the melanocyte on the skin, which express GP100. And sometimes patients feel pain in the tumor. Some patient has a neck tumor, they feel pain. All right, so this is a real medicine. And I think I have a little bit privilege to show you updated information. So this is a very up-to-date information. This is going to be presented at the incoming SMR, but I have a privilege or permission from the company to show. So they still see the significant response, but overall, this medicine makes the tumor stop growing. And then after number 3, number 4, patient is treated as outpatient, and then they just keep coming back and back. So this is very easy medicine after 3 or 4 treatments. And then this is water for plot. As you see, everybody is going to go into the same direction, not the going up. Going up is a progression. We only have a couple patients, fail. So and then phase 1 cohort is almost over. So we found that 75 microgram. Remember, antibody is 100 milligram or 1,000 milligram. So this is much more sensitive. And then choose the 75 microgram because if we go up, patients tend to have a transient liver toxicity because tumor is in the liver. So medicine attack on the cancer in the liver, release the toxin, and then normal liver is damaged, like embolization. So we are okay for that, but the FDA is not okay. So therefore, they are deciding to 75 microgram. Then next cohort is just 2 cohorts. One cohort is expansion of the current phase 1A, and then increase the number of the eye melanoma patient with 75 microgram to confirm toxicity and potential efficacy. And then patient who most likely who fail the immune checkpoint blockade is eligible. And then future cohort is that Dr. Latsky mentioned is treatment naive. And then HLA2 positive. Then pain is, this is a 1-to-2 randomization. We have been discussing it. Company tried to not to do this, but they don't have any good control. So this is a way that we suffer, patient may suffer, at least for short term, but to have medicine available for everybody. So, and if patient is eligible, patient is going to be randomized in dacarbazine or GP100. And there is no crossover at the beginning, but once FDA say, okay, this medicine is working, we are going to give you a tentative approval, everybody can get that, okay? So try to shorten the period of the galbazine infusion for most of the patients. That is about the way to go. This is a clinical trial. So you might not believe, but the person who is given medicine is also suffer. But this is we have to do. Okay. I'm sorry to just late, but this is my team in Philadelphia. Most of them, Dr. Shield. They have a very good eye oncology team, and I'm lucky enough to have all, medical oncology, interventional oncology team. And then Dr. Aplin is a very established basic scientist who is basically working on the MAP kinase pathway and new other pathway, BET1 pathway. So we're working together to make a new treatment available to patients. Thank you.